Thank you very much. I want to thank Dr. Dudwa, who apparently is on his way, but also to thank the Society, the St. Petersburg Society of Naturalists for their wonderful hospitality. It's really been a pleasure being here. Now this afternoon we will witness a rare event. There will be 3 consecutive talks about turtles and how they get their shells. This is wonderful. This is great. I'm thrilled. So I will be talking about an outline of a hypothesis we have concerning how the turtle gets its bony shell. And then Dr. Jacqueline Moustakis will be speaking about her research into how the shell originates and also some of her work on how the scutes, the scales of the shell, originate. And then Dr. Gennady Cherepanov will talk about his research into scute development and evolution. So this should be great. 3 turtle talks. Okay. So again, when we start with evolution, we usually start with Wallace and Darwin, talking about natural selection and variation that exists within a population, and the competition for survival, most animals dying before reproducing in the great and complex battle of life, the survival of those most fit for the environment, and the offspring being from those survivors. And in the 1930s, the new idea that offspring inherit the genes that made their parents fit for the environment. So the notion of natural selection, which explained descent with modification. You have descent, but you also have modification. Okay, well, this could explain the survival of the fittest, but it could not explain the origin of variation. How do you get the variation upon which nature selects, was the question that was brought out earlier today. And of course, Kovalevsky was very important in being one of those people who said that you get changes in evolution by changing development. And by looking at larvae, he was able to unify the entire animal kingdom through cell lineages, larval homologies, and he also actually was able to unify the vertebrates through the bilayered gastrum. And so he was looking at both descent and modification in these embryonic lineages. Now, what was also mentioned today, this morning, was Menshnikov and Kovalevsky looking at evolution changes in development. This was from a letter from Kovalevsky to Menshnikov, and it might be dealing with the changes during gastrulation, the postulated changes. But the first evolutionary biology was really a biology based on evolutionary change. Now, Kovalevsky's research on ascidians and the notion that the ascidian larva was a chordate, it had a notochord, it had and it had other apparatuses that looked like a vertebrate, did not go unnoticed. And Darwin and Huxley mentioned this in their writings, matter of fact, mentioned it prominently. So Darwin in 1874 says, "Thus, if we may rely on embryology, ever the safest guide in classification, it seems that we may have last gained a clue to the source whence the vertebrata were derived." Now, Louis Agassiz, writing the same year, was angry at Kovalevsky's work. He didn't believe in evolution. And so he said, one could hardly open a scientific journal or any popular essay on natural history without meeting some allusion to the Ascidians as our ancestors. Now, one of the people who worked in the same area as Kovalevsky was Thomas Huxley. They both worked on salps, came to similar conclusions, I believe. Now, Huxley here, using Kovalevsky's data, says something fascinating. Well, they both worked on Pyrosonia. Evolution is not a speculation but a fact, and it takes place by epigenesis. Now, Darwin wouldn't have used epigenesis. He would have said it takes place by natural selection. Huxley is using a technical term for development here. Body construction. He's interested in how you get changes that can be selected, how you can get new morphologies, how do you get the arrival of the fittest. But Huxley was writing, 1893, but it really took a century later to get a theory of epigenesis, a theory of body construction. And we see this repeated in something that Huxley's grandson wrote. Here's Julian Huxley. Here's Julian Huxley as a larva. Here's Julian Huxley as an adult. This is Thomas Huxley. Okay? And Julian Huxley, writing in Evolution, the Modern Synthesis, said, a study of the effects of genes during development is as essential for an understanding of evolution as are the study of mutation and that of selection. That's a really remarkable thing to say in the 1940s. The trouble is, we didn't have a theory of body construction even in the 1940s. And so the next slide I'm going to show is for the only slide of real developmental biology that one needs to understand the lecture. And this is, the slide I put in for evolutionary biologists, paleontologists, systematicists. This is the basic structure of developmental biology. Okay. Cell-cell communication is caused by paracrine factors, a soluble compound usually from another cell, meeting its receptor on the cell membrane, on the cell membrane of an adjoining cell. It changes the conformation of the receptor such that a dormant activity is awakened. These receptors, when they're brought together, can make an enzyme. They make an enzyme activity that modifies a particular protein. That protein can modify a transcription factor. There's a lot of chemicals involved here, but basically what they do is they modify a transcription factor which can enter the nucleus and cause a certain gene to be transcribed or to repress a certain gene from being transcribed. But so you get from a factor binding at the cell surface through this cascade to a factor in the nucleus which is able to cause a gene to be transcribed or not. And I'll be talking today about transcription factors and paracrine factors. Now, we did get a theory of body construction started in the 1970s, and Alan Wilson, Mary Claire King, Francois Jacob were involved in formulating or reformulating evolution as tinkering with regulatory genes that make the embryo. These could give you new developmental sources of variation. Around the same time, DNA sequencing was announced, and that allowed DNA sequences to be compared between species, rather than to have to use alleles within a species. And these two ideas, or this idea and this technology, were able to really form evolutionary developmental biology. Now, we heard some of the history of evolutionary developmental biology, and really thank you very much, Roman, for that talk. The notion that evolution results from inherited changes in development, that developmental trajectories can bias which changes are more probable than others, and that a more comprehensive theory of evolution results when the population genetic model of evolutionary change, based on allele frequency, is supplemented with a developmental genetic model based on gene expression. And this is sometimes called the argument from incompleteness, and this is just one example. This is from Hughes and Kaufman, talking about the modifications of the arthropod body plan. It says, to answer the question of the arthropod adaptations by invoking natural selection is correct, but insufficient. The fangs of a centipede and the claws of a lobster accord these organisms a fitness advantage. However, the crux of the mystery is this: from what developmental genetic changes did these novelties arise in the first place? How do you get the fangs of a centipede? How do you get the claws of a lobster so that they can be selected? And Wallace Arthur basically have identified 4, and only 4 ways of tinkering with the genome. You could change the time that a gene is expressed, and we see this in limb size and human brain. That's heterochrony. You could change the place of gene expression. You could change the amount of gene product being made, or you could actually change the gene product itself. So change time, place, amount, or type. Those are the 4 categories in which these changes will occur. So now we're going to be looking at the turtle shell, and this is turtle anatomy for biochemists. There's a large subunit and a small subunit. Okay, the large subunit, the dorsal subunit, is the carapace. This is the dorsal shell. The plastron is the ventral shell. And what you're seeing here are the scutes, which are overlying the bones. So these are the scutes over here, which we'll hear talks about next few talks. But I'm going to talk about the bones. So the turtles have about 59 bones that are not seen in other vertebrates. The bones of the shell, the upper and the lower shell. The whole anatomy of the turtle is wonderfully weird. It's the only vertebrate whose limb skeleton is inside its rib skeleton. It's the only one who does that. It doesn't have a rib cage. And indeed, that's what makes a turtle, is the ribs. So, we're going to look, how does the turtle form the carapace? And the ribs are what forms the carapace. So here we have the carapace, and we're looking at it now from inside the turtle. We're looking up at the carapace. Here is the vertebra over here, and from the vertebra comes the ribs. Here are the ribs coming from the vertebra. But instead of going ventrally and forming a rib cage like they do in us and every other vertebrate, the ribs go laterally. They don't form this concavity. They go out laterally. And as they go out laterally, they enter the dermis. They go into the skin. And so here we have the bones that they form. They form bones. Turtle shell forms bones above each vertebra, and each of the ribs will form a rib. Each of the ribs will form shell around it. There are also bones that form around the ribs. We'll hear more about those later too. So this is the carapace rib. These are the carapace bones. These are the 9 bones of the plastron, the lower shell. You have the 3 anterior epiplastra and endoplastron. And then you have the paired 3, the hyoplastron, hypoplastron, ziphiplastron. But we'll talk now about how are the ribs going into the dermis? Why aren't they doing what they normally do? And then how are they organizing bone to form around them? So here, this is actually from Louis Agassiz's work. Agassiz did not believe in evolution, but he believed in turtles. He wrote a wonderful book about turtles. So here is the turtle embryo, and you can see the formation of the shell here. And what I want to direct your attention to is this area here. This is an area called the carapacial ridge, and only turtles have this. When we look at the cross-section of the turtle embryo, here's the notochord, here's the neural tube. What we see, and here's the limb, is we see this bulge over here. That's the carapacial ridge. You can see there's thickened dermis around it. And here, a few days later, we see again, here's the limb. Limb looks more developed than there. Here's the ridge, and you can see the rib is going into the ridge. Here's the myotome over here. The ribs usually follow the muscles and go ventrally. They go south. But here, the ribs are going into the carapacial ridge. Now, what's doing this? Well, data in the 1970s and 1980s suggested that the ridge, the carapacial ridge, was actually making chemicals that were causing the rib to enter it. They were making chemotactic factors. And this was found by Chester Intema and by Annie Burke And what they did is, in different ways, they removed a part of the carapacial ridge. And then they saw what happened to the ribs. And when Intima took off some of the carapacial ridge, he found that the ribs went into the adjoining carapacial ridge. When Annie Burke put foil between the ridge and the metal foil between the ridge and the ribs went ventrally like they should do in other vertebrates. So it looks like the carapacial ridge was making a chematactic factor that was telling the rib to enter the ridge. Well, we thought we'd look for these factors, and we thought we'd look at FGF 8 and 10, because that ridge looks so much like a limb bud that we had to put the limb in here to show you that's not a limb bud, that's a carapacial ridge. And Annie Burke had hypothesized that this might be a center for activity just like the limb bud was. So it looks so much like a limb bud, we thought, okay, the limb has chematactic factors. It needs to get its skeletal muscle in there. It needs to get skeletal cells to come in. Maybe it's using the same thing for the carapacial ridge. So we looked at FGF10 and FGF8. We cloned the genes, and they were very similar to the ones in chicken. We looked at FGF10 to say, okay, it looks like it's the same gene sequence. Is it made in the same places in the turtle as it is in the chick and the mouse? Yes, it is. It's made in this mesenchyme over here in the limb. It's made in the muscle of the limb. It's made in the genital tubercle over here. We grow these at male temperatures. So it's the genital tubercle. Okay, fine. This is where you'd find it in a mammal. This is where you'd find it in a bird. This is where we find it in turtles. Do we find it in the shell? And the answer is yes. We find it in the carapacial ridge. And this is in situ hybridization over here, showing FGF10 mRNA. So, okay, the first part we got. FGF10 is there. Now, in the limb, there's an interaction between FGF10 and FGF8 to make the limb grow out. Do we find FGF8 in there? Well, here's the embryo, and here's over here the apical ectodermal ridge, which should be full of FGF8 in the limb, and sure enough it is. And then we looked at the carapacial ridge to find FGF8 there, and it wasn't there. And it wasn't there. And it wasn't there again and again and again, no matter what stage we looked at, was not there. So FGF8 doesn't seem to be there, but FGF10 is there, and FGF10 can be a chematactic factor, but it usually needs FGF8 to stay active, to stay on. Okay, so we left this, we went to do other things, and then we had an undergraduate come into the laboratory, and she wanted to work on the project, and we said, great, we're going to do in situ hybridization, we'll teach you how to do in situ hybridizations, and then you can work on something your own. So to test to see whether she could do an in situ hybridization, we had some old embryos. They were too advanced for what we needed to look at. Here, take these, find FGF8 in the AER, and prove to us you can do in situ hybridizations. So she did, and what she found was just beautiful. She found FGF8 at the tips of every rib. That's where the FGF8 is coming from. It's not coming from the ectoderm surrounding the mesoderm. It's coming from the ribs. And so now we have a mechanism for coordinating FGF10, and to make sure that FGF10 is always turned on, the ribs are doing it. So the ribs are helping the carapacial ridge make the molecule that keeps them coming into the carapacial ridge. It really works out very nicely. So we wanted to see if FGF is important in ridge in getting the rib into the ridge. So we grew the embryos basically in organ culture. Okay, we take the ventral part of the embryo off, we take the head off, and so now we just have the dorsum of the embryo. And we put it in culture, and within 2 days we get a very nice carapacial ridge. You can see it over here. We also get an apical ectodermal ridge that you can see over here. That's our internal control. And the ribs go into the ridge. This actually is standing up. It's three-dimensional. It comes off the filter paper. So the ridge is, and the ribs are entering the carapacial ridge. However, if we add an inhibitor of FGF signaling, SU5402, the carapacial ridge, actually, it says there fails to form. It actually forms and then degenerates. And here's the ribs. You can see they're turning. They're going inward. They're going ventrally, just as if they didn't have the signal from the ridge. So it looks like FGFs are providing, fibroblast growth factor, probably FGF10, is providing the signal from the carapacial ridge that tells the ribs to enter it. Indeed, we can use chick embryos, and we can put beads containing FGF10 into this, and when we do, the ribs actually migrate to the source of FGF10 that we put into the chick embryo. And when we put FGF, I'm sorry, this is normal migration. This is with the saline beads. When we put FGF10 containing beads in there, the ribs go to the bead and stop. Okay, they don't go ventrally. Okay. So we're here right now. Ribs migrate into the carapacial ridges because they're attracted to a source of FGF, probably FGF10. And the outgrowth of the ribs and the carapacial ridge can be coordinated by FGF10, FGF8 feedback, just as in the limb. Again, I'm providing an outline here. None of these are completely satisfactorily explained yet. So, hypothesis 3. The ribs act as a signaling center to ossify the dermis. Okay? Remember I said that each bone, each of the major bones of the carapace forms around a rib. And sure enough, when you look at the ribs, they start off as cartilage, but the cartilage turns to bone, and here you see the red. Blue is cartilage, red is bone. You can see over here the bone, and then there's a region of modeling, and then there's cartilage. This is the same shell. Here's cartilage, modeling, bone. So the cartilage is being turned into bone, as happens in all vertebrates. You have endochondral ossification. All of the vertebrates with bony skeleton form their ribs that way. But you could also see these flares of bone elements in the dermis. And if you look at a hatchling turtle, one can see over here that around each rib, there's areas of ossification. It looks like the rib is telling the skin to become bone. And this turtle over here, a few months old, you can see that it's fusing to form the bony plate of the carapace. Now, we went to see what is it that might cause this. And the obvious candidate was a paracrine factor called BMP4. BMP4 is bone morphogenetic protein 4. It's known to form bones. It's also known to be synthesized when the bones undergo endochondral ossification. When the cartilage becomes bone, BMP4 is made. So we were wondering, could BMP4 possibly be able to transform this rather late embryonic, mesoderm, this dermis, into bone? And we got a clue from a particularly awful disease called fibrodysplasia ossificans progressiva. It's a disease of humans, very rare, which causes the lymphocytes to make BMP4. And so wherever there's an infection, wherever there's an inflammation, and the lymphocytes will secrete antibodies they're also secreting BMP4. And as a result, this person gets bones all over their body. And eventually they die because the ribs fuse and they can't undergo, and they can't breathe. So BMP4 is known to cause even adult human dermis to ossify. So we looked to see if the skin, the dermis, was responding to BMPs. And there's a way of doing this by looking at the transcription factor, which is modified by BMP signaling. When BMPs signal, they will modify a SMAD transcription factor by phosphorylating it. So by looking at phosphorylated SMAD1, PS1, we see over here that there's a region right around the periosteum. Here's the rib over here. Here's the rib. This is bone. And you can see this area over here of BMP responsiveness. And you can see over here, here's this kind of tongue of BMP responsiveness. A few slides later, there's bone there. So it appears that the rib is making BMPs that the skin can respond to. Here's a blow-up of another slide. Here's the rib. Here's the bone forming around it. And it looks like there's these cells from the skin are expressing PS1, phosphorylated SMAD1, and are therefore responding to BMPs. So we think that what we have for carapace formation is that FGF10 secreted by the carapacial ridge causes the ribs to migrate into the ridge. The outgrowth of the ridge is coordinated by feedback between FGF10 and FGF8. Each rib serves as a signaling center for the formation of the costal bones of the carapace, and a major signal for costal bone formation is BMP, probably BMP4. So basically what we're saying here is there's heterotopy. There's a heterotopy of gene expression, FGF10, causing a heterotopy of bone. The bone is going to the rib is going to regions where it normally doesn't form, going into a heterotopy here of organ formation, of bone formation. So you have heterotopy of gene expression, heterotopy of rib migration, heterotopy of bone formation. It's forming in places where it hadn't formed before. And that's basically what our notion of, kind of our outline of carapace formation. So now I want to look at how does the turtle get its plastron? How does it get its lower shell, its ventral shell? Here we have the 9 bones of the plastron. Here, again, blue is cartilage, red is bone, and what you should see is that there's no cartilage formation here, that the mesoderm is being changed directly into bone. These are ossification centers. There are 9 ossification centers in the turtle. And if I were to give this to my students, without telling them what it was, just give them like this and say, what is that? I would hope that they say, it's facial bone, or it looks like skull formation, because it's intramembranous ossification. There's no cartilage intermediate, going immediately from dermis to bone. And just when you look at these bones, here's the facial and skull bones of an alligator. Here's the plastron bones of a turtle. They have sutures, very reminiscent of facial bones, skull bones. And so they look like skull bones. Their formation looked like skull bones and facial bones. And we asked, could it be possible that these bones in the plastron of the turtle, could they form from the neural crest, which is where the facial and some of the skull bones come from? Now the neural crest is that area of the embryo which forms the dorsalmost portion of the neural tube. And this is a transient area. The dorsal part of the neural tube undergoes an epithelial to mesenchymal transition, and it puts out these neural crest cells which migrate. And in the skull, you can't see it too well here, they migrate to form the facial cartilage, they migrate to form the jaw, they migrate to form the mandibular maxillary arches. They're really involved in forming the facial skeleton. Again, there's trunk neural crest, which forms the pigment cells, sensory neurons and glia, the adrenal medullary cells, and there's the cranial neural crest cells, which can also form neurons and glia, but in addition can form cartilage and bone. Trunk cannot. Cranial can. So there's a big distinction in the neural crest, whether you are trunk neural crest or cranial neural crest. Cranial neural crest can make bone, trunk cannot. And what we're saying here is kind of heresy. We're saying that the trunk neural crest in the turtle might be able to form bone. Do we have any evidence for this? Well, we did an experiment where we looked at 50-day T. scripta. This is Trachoma scripta, it's a turtle we use. And we looked at the forming bones in the plastron. And so here we've stained for HNK1. HNK1 is a marker for neural crest, but it also marks other things as well. It's actually lymphocytes, human natural killer is what it stands for. It's a type of lymphocyte. But it stains, the classical stain for neural crest, and we see it all around the hypoplastron for infants. And when we do Mallory stains, yup, this is bone over here. When we do a control antibody, we do not get HNK1 staining. So this is HNK1 on the hypoplastron. Here's the endoplastron, the epiplastron. We find HNK1 staining, this neural crest marker, on every bone of the plastron. So we went and we looked to see what's happening with the neural crest, and what we found was that the turtle has a second neural crest. No other animal has seen this. What it looks like is after the original neural crest forms the sensory neurons and the sensory glia and the adrenal medullary cells and the gut enteric neurons and the pigment cells. After all that's done, a second wave of neural crest formation occurs in the turtle. And that wave is so big that you can see it up here. This is a 3-week turtle embryo. It has this is the beginning of it, this is later in the week. It has this huge amount of HNK1 staining. You don't see it in the chick embryo, and just to make sure these aren't pigment cells, we looked at a pigment cell marker, C5, and we see C5 where it should be on the epidermal border. That's fine, these are not pigment cells. These are neural crest cells which are different, which are not found in other vertebrates. And indeed, when we looked at them in a slightly later embryo, here's this band of neural crest cells. No more are being made because you have, the vertebra forming over here. Here are some cells, some neurons, some glia, some ganglia from the original migration. But you also see HNK1 staining in areas going towards the plastron. And here we even see a plastron bone being formed. And it's HNK1 positive. This is staining for neural crest markers. Okay? So these cells also, in addition to having HNK1, show FOXD3. FOXD3 is an early marker of neural crest cells, and we see it on the dorsal aspect of the neural tube. They're also positive for P75. So again, here we see the bone being formed. Here we see HNK1. Here's a condensation with P75. So it looks like they're P75 positive, HNK1 positive. They're also positive for FOXD3, it looks like these are neural crest cells. Could they really be migrating? Let's see if I can get the lights. Can I, is it I was afraid. If I can get the lights down a bit lower. Thank you. What we did is, okay, so di is able to intercalate into cell membranes. It's a lipophilic dye. It only will go into the membranes. It won't go into any of the extracellular area. We put it into the neural tube. So any cell that stains for di had to have been in the neural tube. And when we stain in the chick embryo, yeah, we get a neural tube. And you can see the neural tube there. When we do the same injection in a turtle embryo, we not only get the neural tube stained, we get cells stained around the neural tube, far from it. And indeed, when you look for di staining, one can find it very far away from the neural tube. It looks like cells that used to be in the neural tube have been migrating. And indeed, these are unpublished experiments. When we do this in this turtle embryo here, this is looking at it dorsally. This area here, not these bright things, those are artifacts, but this kind of cloud of cells that's coming down on both sides, those are di positive cells. Those are cells that are entering the plastron, but they used to be in the neural tube. So then we looked at another marker. We looked at a marker which is specific in the neural crest for only being on the cranial neural crest, not on the trunk neural crest. And this is the alpha subunit of the receptor for another paracrine factor, platelet-derived growth factor. So this is the alpha subunit of the receptor for PDGF. And this also is found on other cell types, but for neural crest, it's only in the cranium. And what we found is that it's in the endoplastron, it's in the epiplastron, it's found in the hypoplastron and the zipioplastron. All the plastron bones stained not only with HNK1, but with PDGFR alpha. This means that they're like cranial neural crest. So we did an experiment, again, this is unpublished data, where we took the early stage embryos, which have a working neural crest. And here you can see when we isolated the neural tube, here are the neural crest cells migrating out of the neural tube onto a cover slip covered with fibronectin and lysine, poly-L-lysine, so that these are growing well. They're enjoying their migration onto the poly-D-lysine, okay, HNK1 cells. Here we have a later embryo. This is the embryo in its last stages, where it's making these what we call secondary neural crest cells. And you can see neural crest cells migrating out there too. And they're HNK1 positive cells coming from the neural tube. They don't stain with control serum, but this is the important part. These over here, these neural crest cells do not stain for PDGFR alpha. These do. So it looks like the trunk neural crest is being respecified as cranial neural crest. And cranial neural crest can form bone. So that's our kind of hypothesis that there's being respecification. And indeed, when we look at the dermis, when we look at the dermis of the plastron, we find that the dermis seems to be ectomesenchyme. It seems to have markers from the neural crest cells. Indeed, you can get the dermal cells of the plastron to form nodules in culture, just like facial cells, just like calvarial cells. They look like they are forming skeletogenic nodules. So, it looks like the carapace is formed by heterotopy, the FGF10 expression leading to BMP4 expression, and the plastron formed by heterochromy, allowing skull and facial programs to work in the trunk. Our new projects include testing for the respecification of the late neural crest cells. In other words, are there Hox gene expressed? We know that Hox is expressed differently in trunk and cranial neural crest. And we also want to find the paracrine factors that may cause the ossification of the plastron. And we'll hear about the disputes later. So just want to end with this wonderful quotation from Richard Ellis, that if it weren't for the 250 species of turtles living today, these animals, encased in mobile homes, could easily be viewed as bizarre evolutionary experiments that were ordained to failure. Somehow they did manage to survive, and they're just incredible animals to test out theories of evolution and development. Thank you very much. Thank you. Yes?
I have actually two questions. The first question about the cranial nerve crest, which may break in chicken, and chicken maybe in turtle as well, to the trunk to form attachment sites of muscles. Whether these nerve crest populations are homologous to the populations of the nerve crest which form plastron.
Well, the plastron doesn't seem to, well, we haven't looked to see if there are any muscular attachments to the plastron. The plastron has a bony attachment, which actually comes up and hooks onto the carapace. And so we have not looked at that, except we know that it's HNK1 positive. The notion of the homologies of the trunk of the cranial neural crest in the turtle are really interesting, because there's a bone which I did not speak about in the turtle, which is the nuchal bone. And the nuchal bone is the most anterior bone. And no one knows where it comes from. And there's a hypothesis that Tyler Leeson is trying to get published that says it's the clithron bone, that it's the portion of the scapula that leaves and went dorsally. It's the most dorsal part. And his evidence for all of this has to do with the homology of attachment sites for the cranial musculature, and the neck muscles. And so, yeah, I think that the turtle is going to be very interesting, because it's normal, the fact that it has this appendicular skeleton inside of its ribs, and it has this nuchal bone, which seems to be a scapular element, makes its attachments very strange, but probably homologous to the other vertebrates if one thinks of the nuchal bone as being the clithron of the scapula.
Thank you. And so, am I correct in saying that you are believing in Matsuoka's paper and Matsuoka hypothesis that part of the scapula of mammals and maybe other tetrapods includes clavicle?
I think so, yeah. I think that our data is certainly in agreement with that, yes. Yeah.
Thank you.
Okay.
Thank you. We have one question. Now when we have some explanation of the mechanisms of total shell development, the question that connected to evolution, why do turtles get their shells? What regulatory genes were affected and why?
Yeah. Okay. Now, that's opening the big box, because as some of you may know, there are at least 3 extant cladograms of turtles, and depending on where you believe turtles originate, you get a different story for how the shell forms. If turtles originated in marine environments, then Olivier Riepel has a very interesting hypothesis that says you have to form the plastron first, or at least to get the bony parts of the plastron, before you get the bony parts of the carapace, because that would protect you from predators coming up and eating you from below. If you were on land and you made and you made the plastron first, it wouldn't mean anything. And if you made the carapace first, it would probably mean that you could be rolled over and eaten that way. So, so one big possibility, I think it's probably the right one, is that the plastron developed first, and it developed, as it did in many marine reptiles, as both protection, but also like a keel of a boat for making sure that the tail locomotion makes you go forward. Okay, so it's probably has locomotor properties as well as protective properties. And so after that, the plastron would develop for, the carapace would develop for protection. Now, what the turtles probably already had in place, or the proto-turtle, it probably already had a very thick dermis, which provided protection already. Sort of like the leatherback turtle, which has a very thick dermis, but it doesn't have the costal bones to the effect that the other turtles do. And the first real good turtle skeleton that we have, Odontocalus. Odontocalus has a good plastron, and it looks like it has the ribs in the dermis, in the upper dermis, but the bones haven't formed there yet. Okay, in the carapace. So it looks like that is a probable case. However, there's another group of people who say that there are para-reptiles, what's the one I'm thinking of? What? What?
Everybody knows.
Okay, okay.
So,
No, Udontosaurus, yes. Udontosaurus. Udontosaurus, some people are saying, was a turtle-like reptile, a para-reptile, and that might have not had a marine environment, in which case that whole notion goes away. So, yeah, it's a big question, what the turtle shell was originally for. Now that the turtle has a shell, it can use it not only for protection, it can use it for fat storage, for water storage, all sorts of things. But the original purpose, we really don't know.
Thank you.